All right, I got caught up reminiscing with our Grand Round speaker, and I let the time boundary go a little bit. But so I'm just going to make a few comments, and then I'll introduce Carolyn, Missouri, to introduce our speaker today, who we're thrilled to have come and talk to us. Just want to remind people that next week are the Lussman Award lectures. Every year, the research conducted by the psychiatry residents becomes more and more incredible, and this year continues that tradition. There'll be some wonderful talks. I don't want to spill the beans on who the recipients are, but you should. I hope you'll come and hear the work. It's good to support these young people, and it's I think you'll get a lot out of it. On June 1st, I believe June 1st is the the 100 Years of Women celebration at Yale, and I hope people will come to that. And then on the 8th of June is the psychiatry commencement. And our speaker, the psychiatry residents graduating class, chose Glenn Gabbard, who's a psychoanalyst, to be the graduation speaker, and he'll be speaking on the 8th. So Lussman Awards, 100 Years of Women, and then Glenn Gabbard. So a great lineup for the next few weeks. And Monday is the Yale commencement. And Yale Class Day, which I believe is the day before commencement, is Sunday, which is the day before Monday. Is Hillary Clinton is the speaker, and you might be curious about what she has to say, and of course, that would be a good time and place to find out. So, lots of exciting things happening, and today's Grand Rounds is no exception. Carolyn.
Good morning. Welcome to the Grand Rounds sponsored by the Women's Behavioral Health Division of the Department of Psychiatry. As John said, I'm Carolyn, Missouri. As the director of the division, it's really a pleasure for me to welcome you as members of the department, but also members of other departments and places throughout the university. It's really wonderful to see the integration of different disciplines, skill sets, interests. And in fact, that's really the nature of this particular division. What we're dedicated to is really studying the health of women, studying sex and gender differences, and doing that truly in an interdisciplinary way and bringing people in from different fields to work together to answer real-life questions. And we study a whole variety of things referable to mental health as well as addiction as well as physical health. And one of the great bonuses of being the division head is having the opportunity to invite extraordinary people to come once a year to speak with us about the topic area. And so it's really a great pleasure for me today to introduce our Grand Round speaker, who is an outstanding and highly accomplished researcher in neuroscience who has been at the forefront of studying sex and gender difference. And she really has been somebody who has recognized the importance of looking at the biology of the brain and looking at how that relates to outcomes. And Dr. Goldstein is our speaker today, Dr. Jill Goldstein. So she is a professor of psychiatry and medicine at Harvard Medical School. And she's the founder and the executive director of a brand new initiative that's going to take place at MGH. And it's referred to as Women, Heart, and Brain Global Initiative at Massachusetts General Hospital. And it's a collaborative effort, as I mentioned, that relates to both the MGH and to the T.H. Chan School of Public Health. And they partner with other groups as well to really take on, in a very meaningful way, I believe, the co-occurrence of various disorders. Because I can remember in my training early on, we actually would try to, in studying depression, we would try to find people who didn't have co-occurring anxiety disorders, for example, because that would complicate your sample. And finally we realized that why is this so hard to do? Well, it's because we really should be studying co-occurrence. And the co-occurrence is very great. So among Dr. Goldstein's many honors, she was named the Spinoza Professor by the Academic Medical Center at University of Amsterdam in 2007 for her work on the intersectionality of hormones, sex differences in brain and clinical disorders. And she also has received the Distinguished Scientist Award from NARSAD in 2015. She served on a variety of very influential boards and review groups over time, including NIH scientific review boards, and she's participated in strategic planning at the NIH, in particular with regard to the NIH Office on Research on Women's Health, as well as the Institute of Medicine. As I mentioned, she's an extraordinary clinical neuroscientist and internationally recognized expert in understanding sex differences in health and disease associated with the central nervous system. She's focused on understanding sex differences, first of all, in psychoses. We talked last night about how she trained at a number of different places in New York, including Creedmoor Hospital. And for those of us from that area, recognize that as quite an accomplishment to have worked there. And very interested in how psychoses then relates to other disorders. She studied depression, memory decline with aging, and she's very interested in risk for Alzheimer's disease, as well as this larger issue that I'm mentioning today of comorbidity. As a leader in the field for 30 years, she's been at the vanguard of studying sex and gender difference from an interdisciplinary science perspective, and she has done that in a variety of ways, as I'm alluding to, but in particular, I want to mention the fact that she's also been able to combine technologies, so that she's been very interested in looking at psychophysiology, integrating brain imaging, neuroendocrinology, genetics, markers of immune function, and building larger models for the purpose of increasing our understanding. The current focus of her work is to cross disciplines in tackling the major public health challenge of our time, which is the co-occurrence of depression and heart disease and Alzheimer's disease. And she's doing this by really looking at sex differences to understand the interactions that occur in brain. So all of these things are, in my view, and hopefully in yours, are extraordinary contributions. But one of the things that I'm struck by as I really think about Jill's work is that more than these achievements, contributions, on the face is the quality of her work, the quality and the impact of her findings, and the strength of her commitment in informing the view on the influence of sex and gender on health. So we're thrilled to have you here today, Jill, and it's my pleasure to introduce you to talk about this important concept of shared impact of sex on comorbidity of depression and cardiovascular disease. Welcome.
Well, that's a hard act to follow after that introduction, two introductions, and I'm thrilled to be here. As though your chair was just describing to you, I actually grew up here in New Haven. So Yale has I'm a townie. My father was here at Yale in medicine, and so this is really a big pleasure for me to be here in this capacity. Okay, so now we're going to get started. So I like to start these talks with, like, why is she working on that topic, and what's the rationale? What's the rationale? Major depression and heart disease are the number 1 and 2 primary causes of disability, and that's worldwide. Women, by the way, are twice the risk for major depression, and heart disease, well, that's always been considered a man's disease, but in fact, it's the number 1 killer of women in the United States and in most middle-income countries. And the co-occurrence of depression and cardiovascular disease is really quite high, over 20% prevalence, and it will be the number 1 cause of disability worldwide by 2020. And women are at twice the risk for the comorbidity. In fact, we think that in part, one of the reasons why heart disease is the number 1 killer, aside from the nature of the vasculature differs in men and women, is the fact that there is unrecognized and undiagnosed major depression in many of these cases that go unnoticed. There are no effective treatments for the comorbidity. So it's a major public health challenge. And on top of that, depression and cardiovascular disease and diabetes, cardiometabolic disease, they're independent risk factors for memory decline and Alzheimer's disease years later. Independent of smoking, independent of lots of other risk factors for cardiovascular disease, independent for memory decline and Alzheimer's disease. And Alzheimer's disease has a much higher frequency in women. And we now know it is not only because women live longer. So we have been launched to kind of tackle, my lab's really been focused on this for the last several years, to try to understand comorbidity of these disorders using a sex difference lens, and the sex differences in the shared brain circuitry and the shared physiology that regulates the stress response. Now, stress affects every chronic disease we know, of course, and major depression, cardiovascular disease are certainly no exception to this. So why the stress response? Why circuitry that regulates the stress response? And before I get to that, I just want to say that there are windows of opportunity. There are these naturalistic, wonderful windows of opportunity when the brain and the body are either differentially flooded or depleted of gonadal hormones, and that's in fetal development, in puberty, in pregnancy, and perimenopause and menopause. And we can capitalize on these naturalistic opportunities, which we believe reveal sex differences when they emerge. So for example, sex differences in major depression emerge just post-puberty. But today I'm going to really also focus really on fetal development and what goes on in the development of the brain that places that male or female fetus at a differential risk for the expression of major depression, a long-lasting major depression, and also its comorbidity with cardiovascular risk. So we're looking at the fetal origins. We believe the fetal etiology has to do with sex differences in the development of the brain. It's initiated during the sexual differentiation of the brain, and it involves disruption of the fetal hormonal programming of the brain that leads to sex differences and deficits in specific brain regions that regulate the stress circuitry, that regulate the associated physiology, and even the development of the vasculature. And that leads to mood and cardiac deficits that really get expressed across the lifespan. So what are the developmental risk factors for major depression? Well, there are obstetric complications, things that produce small for gestational age or low birth weight. They're preeclampsia or conditions that produce oxygen deprivation. In fact, the 1954 flu epidemic actually showed that second trimester exposures actually also increase the risk. And finally, the Dutch famine study in the late 50s also showed that exposures during second and third trimester produced higher risks for major depression. And one of the final common pathways that has been discussed around this is the disruption of the hypothalamic pituitary adrenal axis or prenatal stress models of major depression. So, what are the developmental risk factors for cardiovascular disease? In the early 80s, David Barker in England really posed this notion, independent of any knowledge of psychiatric disorders. And he said, what are some of the early indicators of changes in the vasculature early in development that people carry with them throughout the lifespan when later on in life they emerge and are clinically relevant? They're small for gestational age, low birth weight, preeclampsia. The Dutch famine study found this as well. And so many of the same things, many of the same obstetric complications actually also producing risk for cardiovascular disease 50, 60 years later. And again, independent of depression, they felt that one of the final common pathways, and they did animal work around this as well, was the disruption of the HPA circuitry in development, producing the physiology and the vascular changes that led to this notion of prenatal stress models of cardiovascular disease. How about timing? Now, whenever you talk about a sex effect, This is really important for those of you who are working in this arena. Timing is everything. I mean, timing is everything in life, but timing is also everything in whether you're going to find a sex effect or not. And initially, when I first began this work, we took the population level studies and we broke them up into trimesters. And we said, okay, within fetal development, we do know something about sex differences in the development of the brain from animal studies, but here are these population level studies. Let's see which ones produce a sex difference in the risk for these disorders when we just break them down crudely by trimester. And it turned out that second and third trimester was really where we found most of the sex effects and these are population level studies at this point. And it was true for really every model, every pathophysiologic model, whether it was a metabolic model, whether it was an inflammatory model, they all showed a greater probability or propensity to show sex differences in the risk for major depression than in first trimester. Now, first trimester, there were some effects for schizophrenia, autism, but most of the sex differences were occurring during second and third trimester exposures. So what's going on during the development of the brain in second and third trimester? Well, it turns out it's what's called the organizational effects of hormone, gonadal steroids, on fetal brain development. Now, the primary driver of the sexual differentiation of the brain is gonadal hormone-driven sex differences in development. There is sexual differentiation that occurs prior to gonad differentiation, which is direct effects of genes, which can occur in first trimester. But the primary driver are the gonadal hormone regulation of the brain. And in fact, the testes begin to secrete testosterone at the very beginning of second trimester. And testosterone has direct effects on masculinizing the male brain, untrue in rats, but true in humans and monkeys. It also has some indirect effects through its conversion into estradiol by the enzyme aromatase. So again, testosterone, very important in the human brain in masculinizing the male brain beginning in second trimester. And these gonadal hormones, both estradiol and testosterone, are extremely powerful neuromodulators. In fact, they have effects at every level of neuronal growth and development, including neurogenesis, so going to migration, synaptic differentiation, synaptogenesis, and even cell death. They really have effects at every level of neuronal growth and development. This is really important because it's not only in development, it's actually also occurs throughout our lifespan as well. I mean, as we've heard, even in the hippocampus, there's some neurogenesis in the hippocampus, which is very dense with these gonadal hormone receptors. Okay, so a number of years ago, we asked the question from an MRI. I'm a brain imager, so I'm going to show you brain imaging pictures, of course. And we asked the question, we took the animal work, a colleague of mine, my neuroanatomist that I work with, Nikos Makris, we took the literature and we said, okay, what are the brain regions that were really dense with gonadal hormone receptors in the brain? What would be analogous in the human brain, and how would they show greater sexual dimorphism just in the gross size of brain regions in the brain compared to all these other regions that we segmented out in the brain? And in fact, that is exactly what we found, that in this case, it's just red is relatively larger in the female brain and blue is relatively larger in the male brain, because the male brain is 7 to 10% larger, the cerebrum, 7 to 10% larger in the male brain than the female brain. But really, it's relative differences between these regions that really drive functional domains. And in fact, one of the most sexually dimorphic brain regions in the brain, that is, they develop in different ways in the male and female brain, and they really function differently across the lifespan, is what regulates stress and emotion, mood. So what are some of these regions? Now, you're not going to remember some, but you will remember a few that I'm particularly obsessed with. One is the paraventricular nucleus in the hypothalamus, which I think controls just about everything. It's just this amazing set of nerve cells that has so much chemistry in it, and I'm going to tell you a little bit about what the chemistry is. It's the primary regulator of the HPA circuitry, but also the HP gonadal circuitry, the HP thyroid, and lots of other things. And in fact, it's the most highly vascularized brain region in the brain. Not surprising, because it regulates all these hormones to send them out into the body. But of course, so we get arousal in the hypothalamus, we get arousal in the amygdala, we get arousal in the periaqueductal gray and the locus coeruleus, and then we need our cortex, the medial prefrontal, the orbital prefrontal, anterior cingulate, and even the hippocampus to provide that negative, sort of the inhibitory control of arousal. Really important in looking at the circuits, like what causes arousal, and then how do we regulate it. So it's really important for our cortex in particular to help us regulate the stress response. And these regions not only regulate stress and are highly sexually dimorphic, they also are abnormal in major depression and in anxiety disorders, both structurally and functionally. And they also regulate the central autonomic network through this little set of neurocells called the nucleus of the solitary tract in the brainstem that connects directly through the vagus nerve to the heart. So really important in the fight-flight response. Our heart rate goes up, our respiration goes up. We need that cortical region and also the hippocampus to help us regulate that, calm down. And so there's really a whole circuit between how these regions both regulate how our mood and anxiety in response to both positive and negative stressful things, and then how we regulate our heart. And parasympathetic regulation of the heart, that is the inhibitory control of the sympathetic response, is abnormal in major depression, and in particular in women, because premenopausal women actually have better parasympathetic control of the heart than, these are healthy women I'm talking about, than men, age-matched men, and more than postmenopausal women. Okay, so now we have sex differences in these brain regions. They regulate stress. They're disrupted in mood. They're disrupted in anxiety. They regulate cardiac tone. And they also regulate our HPA physiology, as I alluded to before. Paraventricular nucleus is the densest region in the brain with corticotropin-releasing hormone, acting on the pituitary to produce adrenal corticotropin hormone, acting on the adrenal gland to produce cortisol, but also this anti-glucocorticoid effect of dihydroepiandrostenedione, DHEAS, really important in the regulation of stress itself, the DHEAS. Hippocampus providing the negative feedback on the PVN to say, hey, stop producing that CRH, we got to calm down here. And so there's the brain regions also regulate our hormonal response to stress. And by the way, of course, they also regulate the HP gonadal response to stress. And finally, they regulate our immune physiology. So 3 really important pro-inflammatory cytokines, immune factors, tumor necrosis alpha, interleukin-1 beta, and interleukin IL-6 are the primary co-activators of HPA circuitry. So that is, their receptors are densest in the paraventricular nucleus of the hypothalamus and the hippocampus and the pituitary gland. So there's a lot of crosstalk between these gonadal hormone receptors, the glucocorticoid receptors, and the immune receptors, the cytokine receptors. And so they crosstalk with each other to produce both activity in our brain that affects our behavior, but also our physiology that gets sent out, obviously, to the body. And really important is the vagus nerve. This is the other thing I'm obsessed with. If we have a little time, I'd like to talk to you a little bit about a neuromodulation therapeutic we're developing to kind of get at this circuitry. But TNF-alpha and 1 beta are actually released by the vagus nerve, and the vagus nerve, Kevin Tracy has called it, an anti-inflammatory reflex. So it's really important in the regulation of really key pro-inflammatory cytokines that are involved, it turns out, abnormal in major depression and cardiometabolic diseases and in anxiety disorders. These 3 have been specifically identified. Okay. Oh, I'm just going to throw one slide in about memory because there's just too many things to talk about with the brain and the heart. So I'm not going into, and cardiovascular, but another time maybe we can talk about memory function, but I just want to sort of pull that together, and why I mentioned the risk for memory decline is because some of those same brain regions that we study in the stress circuitry also regulate memory function. So, and of course we know that stress affects our memory. I mean, that makes sense, right? Well, there's a biology to it. There's a neurobiology to it. And some of these same brain regions, again, also regulate memory function. So that's why we're really looking at what are some of the shared disruptions of these brain regions early in development that get carried on throughout life, like we have a super immune response and glucocorticoid response to stress, maybe even just minor stress that we carry throughout life. So depending on what environment we're in, it can either be enhanced or get worse or create lots of deleterious effects in the brain and the body, which then affects memory function later on in life as well. Okay, so what is the model we've been working on? We've been looking at, we use these high-risk pregnancies as our model to look at fetal antecedents to the risk for these disorders 20, 30, 40, 50, even 60 years later with sex differences in memory decline, prenatal stress models. And the model is this, that we have been looking at preeclampsia and fetal growth restriction, which changes the maternal immune response. She then has to mount a glucocorticoid response because glucocorticoids have anti-inflammatory effects. So she mounts her glucocorticoid. So there's a disruption of her HPA circuitry, which when it occurs during second and third trimester, will affect the development of the brain in the male and female fetus in a differential way in that circuitry I showed you. And because it's occurring during organizational effects of hormones have some lasting effects on the brain. Like you cannot castrate an animal later on in life and produce the same exact effects in development. Development is really key. Now, I'm not saying that fetal development is deterministic for the rest of our life. Of course, environment either potentiates it or not. Puberty potentiates it or not. But it can set that person on a course that will make it more difficult if they're living in really difficult and socially disadvantaged environments, if they're exposed to trauma of all sorts, if they become obese and have lots of other things happening. So these things sort of can, in the genetic vulnerability context of it, can have these lifelong effects and produce risk for major depression. It's comorbidity with a loss of cardiac tone, which is a major risk factor for cardiovascular disease itself, and that this will be mediated by looking at the circuitry and the physiology. And I'm going to show you some data for this. And I'll just say really quickly, because given that the time we started a little late, I probably won't show the animal studies on this, but I also work with two preclinical investigators of the sexual differentiation of the brain who have really taught me everything I know about sexual differentiation of the brain. They're amazing, Stuart Tobit and Bob Honda. They have looked at both rat and mouse models of this. And so Bob gives glucocorticoid dexamethasone challenges at different periods of development, and then looks at what happens both behaviorally, but also genetically and molecularly in the fetus, in the male and female offspring. And Stuart ran knockout GABA models, because our model has been that there is a disruption of with these glucocorticoid excess, there is a disruption of the gonadal hormone regulation of nerve growth factors like TREC-B and brain-derived nerve growth factor, insulin growth factor. These are two of the ones that we have found results for. We also have some results on even nitric oxide issues, but that it changes the balance of the GABA-glutamate balance, which has changed. Stuart showed the migration of neurons into the paraventricular nucleus in a sex-dependent way. We also actually serendipitously showed sex differences with these exposures and the knockout with the GABA with changes in sex differences in the development of the vasculature itself. So that set us on a course of really looking not just at the cardiac tone through the vagus nerve, but also what was the impact on the vasculature itself. That's it in a really quick nutshell. So if we want to talk later about that, I'm happy. Now, I'm a clinical neuroscientist, so that's it. That's all I'm going to talk about animals. I'm a clinical neuroscientist. It means I'm a brain imager. I like to study people. And how can we do this? How can you study fetal issues, fetal programming, we call it, of things that happen much later in life. You know, NIH is really not going to be funding these sorts of things. And I was really lucky in my career to have access, actually originally with Ming Swang, who was my chair for 17 years. And I've done this work in this prenatal cohort with Steve Buca and the late Larry Seidman. This cohort was established in 1959 to 66. It was the National Collaborative Perinatal Project. In fact, Ty Cannon, I don't even know if Ty is here today, but Ty Cannon also dealt with this in Philadelphia. But there were 2 sites in New England with 17,000 pregnancies, 14,000 at the Brigham, and another 3,000 to 4,000 at Brown. And we have created the New England Family Studies. These mothers were followed through their pregnancy, community sample of mothers, so unselected for psychiatric or medical. So it's a naturalistic sample, community sample of women coming in. They were followed through their pregnancies, systematic data, blood was stored at NIH. The kids were followed for 7 years with just amazing data. These data are available to all of you interested in any of this. We have the full data set. Ty probably does too, but we have the full data set. People are interested in asking questions of this. We are very collaborative, and we want the next generation to take these cohorts into the future because they are a gold mine. And what we have done is, over the last 20 years, we have found the offspring. We have clinically interviewed them. We cognitively assess them. We bring a subsample to the Brain Imaging Center. And we also do extensive cardiometabolic exams. And we now are collecting cells, so we're very interested in immune pathways. And so we do DNA, obviously, but we also collect monocytes, and now we're looking at whether these high-risk pregnancies actually change the innate immunity in a sex-dependent way in the offspring, and that is one of the things that gets carried on through life that really affects the comorbidity in these disorders. So that's really one of the main hypotheses we're looking at now. And we can also do this because the maternal sera is sitting at NIH. It's stored at NICHD. There's maternal sera, there's cord blood, and we've assayed it for these primary HPA coactivators, the immune markers, and some hormonal markers, so that you can actually look at what happens in the mother in the serology from 1959 to 66 and relate it to these offspring and their brain activity and brain issues years later. Okay, I like to sit on that because I'm serious about those of you who might be interested in working with these cohorts. It's really important that the next generation take these over. We are not going to be around that long, but, you know, at least working. You know, we really want these cohorts. This is a gold mine. It's a gold mine, I'm telling you. And I'll show you why. Okay. So we have a very large study of major depression, about 500 cases. Over half of them are recurrent major depression. And we do very detailed clinical assessments. I have super clinical interviewers. They're clinician and also systematic diagnosers. I have gone over every narrative myself, along with one of my colleagues. And these are true major depression. This is not like symptomatized, you know, it's not the DES, it's not symptom screening. They're diagnostically solid, I feel. And then we bring a subsample, about 100 of them. We've done imaging of the stress response circuitry. I'm going to show you a little data from that. We've also done a study of discordant siblings, like you could do these really cool designs. So one sibling gets exposed to preeclampsia and fetal growth restriction, and the other doesn't. Great design for actually looking at what happens later on. And so we've done that with comorbidity, with cardiac tone, because these people are not old enough yet to really have frank cardiovascular disease. They have some hypertension, but not particularly the women, which is later. And then we've even done these recent study of discordant siblings and prenatal stress models for memory decline through the menopausal transition. Okay, So I'm a functional imager, so we induce a kind of negative affective state. I did not want to have any cognitive aspect to this, so you're just lying in the scanner. I'll show you the pictures in a minute. Adapted from the International Affective Picture System. I don't know how many of you are imagers out there, but we do functional imaging. It's really an issue of blood flow. Venous blood has paramagnetic properties that you can pick up in the scanner, and we look at an activation task and a neutral task, and we see the blood, we're really picking up blood flow, but it has been coupled with neuronal activity in animal studies, and hopefully one day we'll get closer and closer to neuronal activity. But really important for these functions is that one brain region really doesn't explain, like the amygdala doesn't explain everything, I'm sorry. It's like there's an obsession with the amygdala here. Okay, now I know I'm obsessed with the hypothalamus, which is harder actually to get a good map for it, but you need a good neuroanatomist, please. Don't just use a computer. Please use a good neuroanatomist. Okay. Anyway, to really get at anterior hypothalamus, and we're not getting to the PVN, but these structures are really important, and just because our computer algorithms do not get to them does not mean that they are not active there. So this is like just a plug for real people looking at these images and creating masks for you. I think there must be imagers here. You guys are such amazing imagers, but so that's my little imaging sermon. But one brain region obviously is not going to explain things. So it's really important to look at connectivity, which we look at with these PPI analyses. And then, of course, we hook them up in the scanner. So we have this tubing, and we hook them up to this stress response, and we get blood. We get blood when they come in. We get blood when they go in the scanner. We get blood so that we get real-time blood assessments in response to the pictures that I'm going to show you in a minute, and we can get their other physiology as well. We hook them up to electrocardiogram, so we can also get their heart rate, and we can look at heart rate variability. We can look at parasympathetic regulation of the heart. While they're viewing the pictures, we can get real-time physiology in response to the brain, and that's what we do. And then we scan the women at different points in the menstrual cycle. Initially, not all the time, because it's very expensive and hard to do, but in our initial studies to actually identify which period we should really, on which we should be focusing. And then we've related these prenatal immune biomarkers to this brain activity and the stress response. Okay, so we show them pictures like this, and we show them pictures like this. So this is a really mild stress. It's not one of your, if there are imagers out there, it's not one of your missed things where they put you under this, like, either your hand goes in cold water or you're under a social stress. It's really not, that's very mild. And you're just lying in the scanner and you're pressing a button just to make sure you're not falling asleep. And in fact, what we did was pretty amazing. We actually activated the stress circuitry, that beautiful stress circuitry. We had beautiful activations in that, and we can actually look at sex differences in that as well. But that little mild task, if you design your study in a way that you know you're going to get internal validity, we were talking about this last night, Carol and I. How do you get that slice so you know that what you see is what you really want to be asking the question about? And so we were able to do this. And I think one of those ways is that you control for the menstrual cycle, which is really, really important when you're interested in really anything. If you take any women into the scanner, you really need to know where she is in her cycle, because it affects brain activity, which I'm going to show you in a minute. So here are women. Initially, this was a study we did many years ago, because there wasn't a study of just healthy. What does the healthy brain do? And is it different in men and women? Not just looking at patients, but really, what's the healthy brain? What's the healthy physiology in the brain? And so we took women in early follicular, we took them in mid-cycle. And we validated this by assays. So we know they're in mid-cycle. So we have blood, initially I didn't have money for blood in 2005, but then we did later on. But what we showed, the same woman scanned twice, counterbalanced, that the stress circuitry actually was attenuated when the same woman was in mid-cycle than compared to when she was in early follicular. Same pictures, scanned about 2 weeks apart, counterbalanced. These women didn't feel any different about these pictures. Everybody's like low, oh, these are no big deal, no big deal. but their brains actually were acting differently under different hormonal conditions to get to the same feeling of subjective stress, which was low, which I thought was just so cool, because no difference can actually be really important. So no difference in behavior, that is. But the brain acts differently to get you to this state of not feeling stressed out when you're looking at these pictures. That's a really important concept. The men look more like women during early follicular. Now, the men also, no difference in behavior. I love this because when I used to give these talks many moons ago about sex differences in the brain, it was like, I'm a 60s child. We were not allowed to talk about sex differences in the brain. And I had a lot of political pushback from the audience, which rightfully so, given certain times. But really important to understand that just because we find sex differences in the brain does not mean it's going to be sex differences in behavior. The brain acts differently under different hormonal conditions. This is really an important concept. The male brain does too, by the way. I mean, testosterone is really powerful neuromodulator. Okay, now, what we found in depression is that there was a hyperactivity in those arousal regions. And by the way, this, let me just say, this difference was driven by estradiol. In a subsequent study we showed this difference was driven by estradiol, not by progesterone, 12 for progesterone. Although we didn't have all the metabolites of progesterone, but like allopregnanolone and things like that. But still, this was primarily driven by estradiol. And then what we found in major depression, which is true of major depression, that there was not a regulation, so there was a dysregulation of the HPG circuitry, which did not regulate estradiol through the menstrual cycle. So the activation, this hyperactivation in the female brain was, and these are, you can't read these, but these are just these regions. That's why I made them very large, and they're right and left. And here's the woman in a sort of the middle, the beginning of her cycle, and here's mid-cycle. And you can just see that here, there's really no difference between when she was, where she was at in her cycle. But in the healthy women, you see a big difference in the activation of these regions if she's in a high estradiol state or a lower estradiol state. So there's a dysregulation of HPG. Now, what about how the brain regulates the response to stress? Okay, so here's some of our connectivity analyses. In this case, I'll just show you, here's the amygdala, so you get the arousal in the amygdala. It's calling on the orbital frontal cortex to actually help to regulate that arousal. And this is just, if the number's higher, it means the connectivity is stronger. And you can see here, here's dysphoric mood on a scale. And you can just see here, this is just represented, these are women, these are men, that depending on the mood, it really didn't matter how connected these were. It didn't affect the connectivity in the male brain. But as the woman became more depressed, right, more depressed, there was less connectivity in the female brain. And that was true both for the hypothalamus and also for the hippocampal regulation on the amygdala. So in all three cases, it was really the female brain that had more trouble regulating mood and the stress response, negative stress response, when she was more depressed. So we have a dysregulation of HPG. We had hypercortisolemia was also related to that hyperactivity, so HPA deficits in major depression. I don't have time to go through all the data, so I'm just kind of going to summarize it for you because I want to move on to really showing, having a little bit of time to show you the prenatal exposure, and I want to leave time for questions. But there was a significant loss of cardiac tone, so the parasympathetic regulation of the heart was very low. In people with major depression, these were people with recurrent major depression, and by the way, they were unmedicated. This is the other thing about doing studies in a community sample, which 60% of people don't get into treatment with major depression. So these were recurrent major depression, people with recurrent major depression. They were primarily, except for about 3, unmedicated. So we're really getting, and they were in remission when we did this. So I really was interested in what's the traits that are involved and unconfounded by medication. That was the question in this study. So loss of cardiac tone was also significantly related to those brain activity deficits. So again, remember the slides I showed you at the beginning where you've got the brain regions, they're regulating mood, they regulate anxiety, they regulate the heart, they regulate the physiology. So this is just data to show you that there's a confluence of things that happen in the brain, but also of course, the physiology is all the physiology that goes into the body as well, and regulating the heart. And of course, we found these sex differences. Okay, so now we want to just really go for what were the immune response in the mother. These were people that primarily, these high-risk pregnancies were related to her change in the immune response. So we're now looking at these assays I just showed you for these three pro-inflammatory cytokines. And we're looking at it in relation to depression and the risk for cardiovascular disease, because they're too young in this sample to go for the disease itself. So it's loss of cardiac tone or a low high-frequency component of the RR interval of the heart. So that's heart rate variability for those of you who do this. And the high-frequency component gets at parasympathetic regulation. And we think this was mediated by those neural circuitry deficits I showed you and the physiology deficits I showed you. Okay, so again, here's the maternal cytokine during pregnancy. This particular assay was taken at the very beginning of third trimester, but we've also looked at cumulative effects in both second and third trimester, and actually cumulative is really a nicer marker of what's happening, or what can affect the fetal brain. And what we found for those people with recurrent major depression is that the odds ratio, really TNF-alpha, and TNF-alpha in relation to the anti-inflammatory effect of interleukin-10, which is an anti-inflammatory cytokine, was really important in the relative risks for major depression, and differentially in men and women. Now, men obviously have major depression, so there are things that affect them too. I'm not saying it's only women. But what was interesting here is that there's really a difference. And we found a lower exposure of TNF-alpha in the mother, predicting risk in the female offspring. Now, people would say, oh, wow, what's that about? But it turns out if you look at the prenatal cortisol that's associated with that, so remember I told you glucocorticoids have anti-inflammatory effects, so you can't just look at one thing. You really have to look at the whole confluence of what happens biologically. And so when we looked at that, of course, the people with the mothers at the low TNF-alpha at the point of really looking at low exposure, turns out had higher prenatal cortisol. So they had lower TNF-alpha because we were just getting a one-size shot in third trimester. So this is why it's really good to kind of look at these things over time and so on. That would be really nice if we had all those data, but you take what you can get, which I think this was pretty amazing that we even have this. And then you get you get really a positive ratio in the male brain. And in fact, the same thing produced low high-frequency component of the RR interval, so a low cardiac tone. So the same exposures in the mother produced risk for major depression in a differential way in the male and female fetus. This is 45 years later. And also a lower cardiac tone, so dysregulation of parasympathetic. And this was this was really sort of so something, this was telling us, of course, something gets retained across the lifespan. And then we asked the question whether it was mediated by the brain. And in fact, what we found is that it was mediated both by hypothalamic, and this was anterior hypothalamus, which includes the PVN, hypothalamic dysregulation, and in relation to those exposures, and also hippocampal dysregulation. And so this finding, let me just kind of go back, this finding of the low exposure or the ratio in the men was related, so the low exposure of the TNF-alpha was related to hyperactivity in the female, and it was also related to hyperactivity in the male, but a little less so. And here we find the interaction that we found with the risk for the disorder, such that the hippocampus, hyperactivity in the hippocampus with low exposure of the ratio, and the opposite for the men. So just showing you that interaction, both producing risk for the illness, was associated with deficits in the brain, mediating that, which get retained across the lifespan. Okay, I'm going to skip the animal work because I really want to just show you one other thing we're doing, and then I'd like to stop for questions. At this stage in my career, as Carolyn told you, I'm launching this new initiative. I have got another vision push in me. And what's really important to me after doing, many years, probably, over 30 years of work in this arena is to translate it, is to actually do something useful. I feel like, make something like a product, something that patients can use, and to make an impact in the fact that today, in this day and age, where everybody's claiming precision medicine for crying out loud, that they do not consider the sex of the case, of the patient. This is like unreal in medicine. It's just really not good. And Harvard and Yale should be at the forefront of incorporating what we know now. We know stuff now. We know stuff. We really know stuff clinically now about sex differences in medicine. Okay, why do you think women are dying from heart disease, right? One of the things is the vasculature. I mean, we know the differences in the vascular. Cardiovascular disease in women is a microvascular disease. The stress tests that are used are not indicative of microvascular disease. So women are getting picked up when they're much more severe, and they're dying of a heart attack, an MI, at a much higher rate than men. This is unacceptable. Okay, you've already heard this from Carolyn, but because this is the lecture for women's health and sex differences, and women's health, we think, obviously, hopefully now, this group understands this is not just about reproduction. It's about really every chronic disease we know has a sex difference in this, and it should be incorporated into medicine. So I'm going to try to do my part in this and use some of the information and keep enhancing information and knowledge, which is really important around markers, but really try to intervene early, because we think some of the same things that are happening in development. Depression emerges prior to cardiovascular disease itself, but we think some of the same pathways, immune pathways, gonadal hormone pathways, genetic vulnerability issues, vascular and metabolic function, that these pathways are shared between the brain and the body, and that if we can intervene early, maybe even just post-puberty would be great. That would be great. Then we can start to actually prevent or at least attenuate disability later in life with these disorders. And one way that we are, so taking a life course perspective, really important. And, oops, shoot. Okay, not done yet. One way we're going to do this, I'm just going to take a few minutes, is really to try to attack the problem of major depression and its comorbidity with hypertension, and to do this in a sex-dependent way. But I'll just show you a little bit about what we're developing, because I think it's so cool. And also when, you work with really, bioengineers, young people who really are very techie, as opposed to me, it's really cool to take things that you've done, and they're developing these devices, and we've really become very interested. Of course, I've been really interested and done a lot of discovery work around the vagus nerve, which does act differently in men and women. And it's the major conduit between the brain and the body, the electrical conduit. So it's really important. And by the way, it's not just, of course, to the heart, but of course it's the vagus nerve. So it wanders, it goes into the gut, and so it has a lot of implications for immune physiology and response in the gut, and so on. Okay. Now, what we have done, there's a branch of the vagus nerve called the auricular branch of the vagus nerve, and my colleague developed a device. Vitaly Napadou was a neuroscientist and engineer, And he developed a device which is really kind of two acupuncture points in the ear, where you stimulate these 2 points in the ear, and that stimulation really goes directly to the auricular branch in the vagus nerve. Now, the novelty that we have added to this, which was really a discovery of his about 10 years ago, working in pain, but now we're applying it to major depression and to hypertension, is the fact that we enhance the activity of this, it's called transcutaneous vagal nerve stimulation, with your own respiration. And we have enhanced responses with expiration versus inspiration. So we are using this in people that are undergoing major depression. And what we found, we're mapping out the circuitry associated with it, so we can activate that little NTS I told you about, the nucleus of the solitary tract. And now we're mapping out the rest of brain circuitry involved, particularly how it's directly connected with the PVN, this set of nerve cells. We're doing this in a 7T, so it's really great. You can really kind of see, and we can do it in some real time. And look also at other things around blood flow. And what we have found, just in an initial study, we really need to be moving this forward, which we are. But we have found in major depression that we can lower anxiety, we can lower depressive mood, raise depressive mood. And we can also regulate cardiac tone at the same time. Increase the parasympathetic regulation of the heart, and now we're showing that we can also lower blood pressure just by this stimulation in the ear to get at the vagus nerve. Now, those, I'm talking to psychiatrists, so you may know about implanted vagus nerve stimulation, which is used for treatment-resistant major depression, but of course this is transcutaneous, which means no surgery and no drugs. So we think this can have, we are getting an acute response in these samples that we're looking at. And that also may have some really wonderful, if this holds over time, and we can really get an acute response. The only other antidepressant we know that has an acute response is ketamine. So this could be huge for even an adjunctive potential therapeutic. And we are going to develop it in a sex-dependent way. Of course, I'm putting that out there. Okay. And that is one of the goals of this new initiative that I have launched called the Women, Heart, and Brain Global Initiative. It's focused on these 3 chronic diseases using this sex differences lens through the lifespan, as I showed you. It is a collaboration of Mass General with the Harvard School of Public Health, both the Department of Epidemiology, but also the Women in Health Initiative, where Anna Langer is a global policy expert. And globally, in low and middle income countries, they are really concerned with non-communicable disease. So we're partnering with some global partners as well around this. And finally, this kind of work takes a village. So I'm very grateful to a very large team of wonderful people who work on these studies, finding people 20, 30, 40, 50 years later, engaging them, having them come to the imaging center. This is not an easy task. This is not for wimps. I mean, this is a marathon. I just have these great colleagues. As I said, these are the animal investigators I work with. I work with some people at Columbia, and I'm grateful to NIH for funding our work for about 30 years. So thank you.